Okay. Hello!
Hi there.
Welcome. Welcome to Microbiology Journal Club, where we nerd out big about all things small.
My name is Danny, and in a previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based at NYC dedicated to aid the public with the tools of biotech.
My name is Faz. I have a PhD in microbiology. I've mostly worked on bacteria, but I've also worked as a research integrity specialist. I'm currently working as an editor for a scientific journal.
Each week, we meet to talk about microbiology, and today we're doing an overview of some of the coolest microbiology papers, a lot of Omicron, that we've seen this week.
And at the end of the month, we'll be selecting a paper to do a deep dive on, so make sure you message us at microtwjc if anything has caught your fancy.
You can follow along with the papers that we discuss in either week in our shared Zotero library, linked in the doobly-doo below.
And we want to hear from you, so please use the comments or tweet us at microtwjc. And boy, do we have a show for you today. We're looking at a bunch of Omicron preprints, analyzing how Omicron infects cells, and you get some clues as to what makes it different from other SARS-CoV-2 variants that we've seen before. Not only that, we also take a look at the immune responses, how T cells react to Omicron, and you also look at the potential for SARS-CoV-2 to travel all over the body during an infection, including in the brain. But wait, there's more. We learn about how salmonella transmits between host cells by forcing them to eat each other effectively. And finally, we look at the genes in eukaryotes, like us, that we have stolen from viruses. It'll make sense-ish when we get to it.
Yeah, and so to kick things off, we have this first Omicron paper. SARS-CoV-2 Omicron variant shows less efficient replication and fusion activity when compared with Delta variant in TMPR SS2 expressed cells.
So yeah, this is following on from a story we've been following a lot. So for those of you who might not be familiar, spike protein is the protein of the SARS-CoV-2 that binds to ACE2 on the surface of cells. But in order for the spike to actually allow the virus to fuse with the membrane of the cells, it needs to to form this fusion combination. It needs to change shape, but that only happens when it encounters a host cell protease like TMPR SS2. And so that cuts the furin cleavage site and causes a spike protein to transform and fuse with the membrane.
Yeah, it like
But
opens up like a
yeah,
flexible part that like then when it comes into contact, yeah, it'll fuse that membrane. And that
yeah,
TMPR SS2, that's expressed on the surface of different types of cells. So the thought process is like spike allows these viral particles to come into close contact with the cell, then where it encounters some protease, or if it previously encountered a protease that was just floating around, that could also be the thing that happens, and then it'll fuse right at that moment. It grabs ACE2.
yeah. Delta was very good with fusion. I think we saw like with the P681R like mutation, it fused very easily. Whereas Omicron, even though like it's that site where furin cleavage happened, it seems to have some mutations that would benefit it. Actually, it seems to not be cleaved very well at all. So we've seen that in other studies. I think we see it here. So this one tests the spread of Omicron using Vero TMPRSS2. So these are African green monkey cells that are engineered to express TMPRSS2, and they have ACE2 on their surface. So this way you can test compare Omicron and Delta, see who like can infect these types of cell better. And Omicron tends to do much worse. Um, so let's see if I can pull up a graph quickly.
Yeah,
To
one of the first figures.
yeah.
Yeah.
So what you tend to find is that when there's TMPRSS2 present, Delta does better than Omicron. And when you take that away, they do kind of equally well-ish. And in lung cancer cells, so CALU3, Delta does much better than Omicron.
Yeah, and like we had seen one of the things we saw too was like in those ex vivo cell cultures, right? That like Delta was doing better in lung cells. Like this matches with the CALU3 data that we're seeing here.
Yeah, that matches with what we would expect. But that is still like kind of a mystery because Omicron is spreading much faster than the Delta variant.
Right.
So, which doesn't kind of in my mind, I'm thinking, okay, well, if it infects cells less easily than Delta, then why is it transmitting so well? And that's kind of the the detail that we want to get more information about, I think.
Yeah, that's the big question that hopefully some of these studies are helping to tease out a little bit. Um,
Yeah. So
and
yeah,
so,
we're
oh, I just want to say, so the way they, at least the way that they're going to try to address it here is, and this, this is a big, this is reminding me of like when we first talked about TMPRSS2, like I believe I was quite confused, and just because like they were using two inhibitors, one to inhibit this particular cleavage, but then they are also using another inhibitor, which comes up here as well, that inhibits like another type of cell fusion event that would happen in the endosome. So instead of like
yes.
fusing right at the surface of the cell, where it hits the receptor and then it initiates whatever needs to happen, basically like think about like fat bubbles or in your vinaigrette, right? The vinaigrette bubbles like merging. This is kind of like a similar idea in viral fusion, but
Yeah.
the
Intelligent fat
other
bubbles is what I'd call them.
yeah, yeah. But the other place that it could happen is there's like an endocytic pathway where cells are sampling things from the environment. So you might hit a particular type of cell or a receptor type, and it'll actually just grab whatever is attached to it and bring it into the cell. And of course, now it's in an endosome, so lots of other things can happen in an endosome, but fusion is one of the outcomes there, and that could be another way into the cell. So they have an inhibitor here, camostat, right? And
Yeah.
camostat inhibits the endocytic part. Is that correct?
No, I believe Camstat is the one that inhibits the TMPRSS2 cleavage. So
Okay.
that was used in
Oh,
the
so
previous
this yeah,
study.
so this is inhibiting this surface-based internalization.
Yes. But they use other inhibitors to inhibit the endocytic pathway. So I think that would be that. Let me check my notes
Oh,
here. So
chloroquine.
chloroquine, yeah, and
Yeah.
baphylimycin A1. So they use these
Hmm.
two different inhibitors to try and test whether that changes. And with some of them, actually, like hold on, yeah. So com- so yeah, some of them they actually changed the so like when you like say knock down the TMPRS2, Omicron does much better. Whereas if you add in chloroquine, there isn't too much of a difference by the looks of it. It's a
Yeah, what are they
so
in A? They're using baphylimycin A. What are you
A1,
looking?
and in B they're using chloroquine, and it just basically seems like there isn't too much difference between them.
Hmm.
So Chemistat only worked well against Delta. Um, yeah, this kind of provides a little hint. Well, they're trying to ease towards this idea that the endocytic pathway might be playing a role in Omicron. Um, yeah, they look at the fusion activity, and yeah, so this kind of like hints towards the idea that, and the reason why they might be thinking this because other related SARS-CoV viruses are primarily use the endocytic pathway rather than um, the furin cleavage pathway, if I recall correctly.
Yeah,
Um,
the endocytic pathway.
yeah, because I think like the basically like the furin cleavage pathway is like the killer app for SARS-CoV-2. That's the thing that
Mm.
really made it much more transmissible. And
Hmm.
so they naturally, if they're thinking, okay, Omicron might be spreading a different way, so let's look down at other options. So that brings me to the next paper, not my
Yeah.
paper. These are not related to me.
Oh, you weren't like fifth author on this one?
No. I think I like only know like one of the authors. I think maybe, but that's through like a some tangentially related via like lab contacts. Um,
Yeah, yeah.
but
Yeah,
yeah,
so
so
in this one, so this one's called the hyper-transmissible SARS-CoV-2 Omicron variant exhibits significant antigenic change, vaccine escape, and a switch in cell entry mechanism. So they're gonna
yeah,
also overview in this paper. They also overview some stuff that we've talked about before, like oh, let's look at all the mutations across the Omicron spike, and like let's look at how it's related to the rest of the strains.
yeah. So I'm probably going to try and skip over that because we've been through quite a lot of those sorts of
We
details
have, we
before.
have.
Um,
I do want to point out that in their map, that's like 1C, in figure 1C, they have that map. And, we've heard this story before, Omicron, it's like, has all these mutations, and people are wondering, like, where did they acquire these, because we haven't had that surveillance. And at the root of the tree, they have OG, which stands for original genotype, just so everyone knows.
Oh, original genotype. Oh, I thought it was original gangster. That both make sense.
That's at the root of this tree.
Yeah. So
Oh, yeah,
obviously
they have antibody data in this one as well. So, that is with previously vaccinated individuals, previously reinfected people. Is there any difference in the way we've seen some of this before? Is there any difference? Of course, things are not as reactive with Omicron as they are with others. Oh wait, this is all Omicron. So they're comparing across the different vaccines here. So
yeah.
all the vaccines are kind of the same.
Yeah, I mean, I feel like the AstraZeneca one tends not to do as well. I feel like there might be a vaccine waning effect, but again, I'm not sure anymore. Now that so much data's come out, I feel like someone would have accounted for it or have answered that question.
Would have said it.
But one thing but one thing like I do like about this is the fact they have a control like from the same people for like the N protein, which is not something I've seen before, and I think it like puts things in perspective that even though there is vaccine waning, it isn't like no protection like you'd see with the N.
That's a great point.
Let's see, but they also focus in on the idea of the endocytic part of the switch cell entry mechanism.
Oh, but we see they also go over the booster. So I guess I'm running through all these figures just like to reinforce for people. We're seeing a lot of this data like before it's being collected nicely for us. Boosters do boost your antibody titer.
Yeah. This is, I probably should mention, this is a preprint where it's not even in a preprint server, it's just still under. So this is very much like an uncooked, like half, I don't want to say half-baked, that's got bad connotations, but it hasn't been fully
they're still
peer-reviewed or
the
formatted yet.
They probably had, right, like, because as we've experienced on our show, right, like the news is like super fast in where it comes out, and scientists are working in their labs, like making this data, but the traditional publication method is like a very long, comprehensive story about what's going on. And like this is very much this lab's, or the collection of this group's comprehensive story about Omicron. And so like we're seeing a lot of that same stuff, their data to a lot of the same points that we've heard in the news. But yeah, we're trying, Faz, you want to highlight something more about this alternative entry section of the.
Yeah, that's that's basically the bit that caught my eye. Because I feel like this report is is more directed towards like government or people, like because it feels like an update on every single story rather than just focusing on one interesting part. So one thing is like Omicron doesn't induce cell syncytia, so that's usually characteristic of very fusogenic viruses that confuse a lot. So that again adds to that kind of story. Also, they saw reduced replication in lung epithelium cells.
I want to say that these
that's still something.
Images, though, they might be like really like last week we talked about being less fusogenic too, and they had like worked out some transwell method to like make it known. But I like the pictures here because you can really see like Delta is very fusogenic. Like those cells are kind of a mess of things all like mashed together.
Yeah, and you see like there is a step change between the like I think this is for the alpha variant, like a step change in that level. So
yeah, like part of Delta's
paint a
success
picture.
was the ability, like whether or not this happens in disease or not, like this is like a feature of its spike protein.
Yeah, and Omicron, it's baffling, like how different it looks.
It's taken a different evolutionary path. I think that's one of the reinforcements that I'm feeling or I'm thinking about as I look at this data. It has a different path to success.
Yes, there is something different about it, and in here this paper they do talk about how they think it's because it switched the way it enters into cells. And so as a control, this is interesting. So they use a pangolin coronavirus spike as a control so I think they use like they express these in an HIV, not an HIV virus, a lentivirus perhaps.
Lentivirus, yeah.
Um, and then they tested out all these inhibitors to essentially, because this is for the pangolin virus, they know that goes through the endocytic pathway, so that's why they use that as a control to see whether Omicron behaves like that, more like that compared to the other SARS-CoV-2 strains.
Yeah.
So they do some fusion studies, and I think they basically come to the conclusion that it seems to behave a lot more like those pangolin than
Yeah.
it does
Actually, so
to.
this is great too. This is in figure A, they give us the overview of their hypothesis, right? And this is again like something that confused me when we were originally talking about the fusion, like way back when, many times. So if you guys go back to that episode, like you'll just see me kind of stumble. Maybe both of us stumbled.
Yeah, no,
on
I
this
definitely
idea.
stumbled as well. This was because back then we were just still learning about everything, so we're just learning the lingo about lots of things. Now I've heard
Yeah.
Camostat a number of times, and I've heard about E64D a couple of times, so now I feel like a
Right.
lot more confident about
Yeah.
understanding this sort of stuff.
And here it's laid out really nicely, right? There's two pathways potentially, right? Using ACE2, but on the cellular side, it's requiring different factors. And they have an inhibitor. This is just a drug that they have, and that allows us to study it because you can add that drug and you can block, right, one of those pathways over the other. And so if you see cells, aren't getting infected, in this case by their pseudotype lentiviral vector or whatever, then you know that like that's one of the pathways that those particular viruses are relying on.
Yeah, and they also they use different cell types and I think that kind of builds the idea that certain cell types might be more resistant to entry via the endosome. So,
Yeah.
like for instance, the Calu3 cells, which are I think derived from lung carcinomas, that they might be more resistant compared to say human embryonic kidney cells. Um, so I think that's an interesting point and again they add in. So I think this relative in D, I think that paints the picture about the similarity in infection between the pangolin and the Omicron. Because adding Camostat doesn't seem to like decrease or have an effect on the infection that much, but adding in the endocytosis inhibitor completely knocks them down, whilst the delta is unchanged.
Yeah. And so this is in vitro. These are considered in
Yeah.
vitro studies. They're using
Yes.
cell types, and again, going back to thinking like, what is this? Like, how are they accessing this data? It's like they have cell types that have all types of ways of getting things in, but the manipulator is knowing that they can disrupt one of them over the other. And so, like in this toy example, like you can really tease out, I guess, what pathways might be being used. But of
Yeah.
course,
I mean, I
sorry,
think.
but of course, in the
Yeah.
body,
No.
there's going to be like many cell types, and so like this data could also tell us, like it's also telling us that, like in that complexity, in that complex instance where you have many different cell types, like it's going to change, right? Like the tissue tropism is going to change based on this sort of idea because different cells will align with these different types in different ways.
Yeah, and of course these are using like kind of domesticated cell types, cell culture, cells that we know work well in culture. And I think that there is always some asterisks for these sorts of studies because again, cell culture doesn't the actual human body is a bit more complicated than these models, but these models can give us an idea. So
Yeah.
what we're doing is almost like circling the hypothesis. We've got and every time we add more data, that kind of narrows down to what's actually happening.
Yeah, it makes people
And
feel more confident that they're going down the right track. Like, right, like this data it fails to disprove, right? Like the theory that's being forwarded, and so we can try to use that. We can try to get closer to that by using a closer model next, and we wouldn't have expended those resources, right? Like of trying to do primary cell culture or like trying to find that needle in a haystack inside of like a really large human population and then longitudinal observational data.
yeah, I mean, theoretically, you test all the cell types we've ever discovered, but I think what we're doing is then trying to like pick out a hypothesis and use that to direct and figure out what's going on.
Yeah.
I'm thinking after reading this, I thought, well, what we've seen so far is people testing things in the bronchus. What we've seen is people testing things in lungs. But what actually happens in the nose, the nasal epithelium? Because that's going to be that's a really important part. If you've got this theory that Omicron is more infectious in the nose and upper respiratory tract, I really want to see some evidence to indicate that.
Yep.
And
that
I felt
brings
like
us right to our next.
yeah, because up until like today, that was gonna be my my note of skepticism. Then this preprint appeared to me yesterday, and so I had again, this is a preprint. Like it was someone. So one of the researchers shared this on their Google Drive. Um, again, I'm not bitter, but this is one of the people one of these people is from work. I remember like I tried to do a project with one of these people back when I was an undergrad, and they refused. So I'm not salty about that, but a little bit salty. But yeah,
people are busy. You only have so much time.
I
in the
get
day.
rejected by the best. But yeah, but this is a great like addition to this kind of idea, which is the SARS-CoV-2 variant Omicron shows rapid replication in human primary nasal epithelial cultures and efficiently uses the endosomal route of entry.
So just like in the previous studies, they have Calu3 cells, they have Vero ACE2 TMPRSS2 cells, but they have another cell type here, these human nasal epithelial cells.
Yes, very interesting. Yeah, so and they test them out. So let's see, because I think we're basically familiar with the idea of. So they've got these nasal epithelial cells, and they have. Let's zoom in a bit. I hate. Okay, right. So
Nice.
yeah. So what we have is the Omicron, which is in purple, and Delta, which is in green. And so what they find is in human nasal epithelial cells, it seems that like at 24 hours post inoculation, like Omicron actually has a boost. It seems to be competing. It seems to be doing much better in affecting those cells at an earlier time point. It seems to work quicker compared to Delta. But although Delta in the end seems to be higher. Although what the authors basically say at this point is that the reason for that is because Omicron started killing off the cells in the culture, and that's why it started to decrease. Because when you kill off cells, less virus factories, whereas Delta, like those cells were still alive. And whereas like
yeah, because in the match in that same in the three figures underneath, that's PFU. So first they're measuring just like in the top 3 grams at E gene copies. So that's like replication amount. They're just seeing like how much DNA is being made inside of these cells. But underneath that they have PFU. That's like then how many viral particles came out after they you take out the liquid and you count how many particles are in there by like a plaque assay, something like this.
Yeah, and when you look at that, it doesn't look so good for Omicron. Like you see that increase at 24 hours, but then like there are less viruses. But if you've got that controller rate in terms of the ratio, if they were able to adjust for the death rate of the cells, perhaps that would make this a bit more of a convincing graph.
Right. That's like one of the confounders that we're not sort of seeing in this data set, right? We're not seeing like how much did the cells die normalized against that in any way.
Yeah, and now thinking about that, I'm going to start thinking about that every time I see cell culture data now. I think this is...
Yeah, because it can, right? Like you can paint a story that looks one way, but like actually in the dish it might be different because, we're not looking at the dishes. We don't have that like rich set of experiences. We have to rely on the authors to show it to us.
Yeah. So what they did next, I think, was they did like a head-to-head. So this is a competition assay where you put 2 viruses in the same petri dish and see which one wins.
Yeah, cage match.
And yeah, in the previous study they did that in the Vero ACE2 cells, and they found that the Delta tended to win out. Whereas like here, when they use the nasal epithelial cell cultures, Omicron tends to win out at the end.
Yes, and they still replicate that in Vero cells, like Delta. Well, I guess it's kind of skewed, but like there's no clear winner in those versus in human nasal epithelial cells. So there's definitely an opportunity, right? This niche is more preferred in these experimental conditions.
Yeah, I mean, competition assays can be tough. I mean, especially if they're if the competition is sensitive to initial conditions, and if you're counting, like, say, thousands of viruses, there is a margin of error sometimes that can mess. Because I remember I did competition essays for when I was like during my PhD, and
Yeah.
they were quite difficult to make sure you get everything even. Um,
Hmm.
but
And also sometimes it matters. So like in these two, they did different ratios, right? A, they're using 50-50, but in B, I think they use more Omicron to begin with, 8-2.
yeah, and I think that's kind of when you skew it in the favor of like one, then it's more impressive when you suddenly see that Delta takes over in the Vero Ace 2 ones. When you start off with, oh, it's very low, or
Yeah,
even
or in
the
the Calu threes.
yeah.
Like yeah, so like it gives you that, it gives you more confidence that you can say that in Calu 3 cell type, like Omicron is at a disadvantage compared to Delta for sure. And but in human nasal epithelial
Hmm.
cells, Omicron's at an advantage.
Yeah.
I guess they could have
Um,
done they could have skewed
so
it, right? To make it feel, to really emphasize the advantage in the human nasal epithelial cells. It'd be nice to see the 8-2 with the Delta strain.
I mean, I can imagine, I can understand why they didn't do the. I mean, actually, yeah, that would be interesting if they did that in the because then that would like strengthen the idea that Omicron can actually
Yeah,
outcompete Delta.
it has a real
Um,
advantage in human nasal epithelial cells.
yeah.
Okay. Anyways.
So that would. So yeah. So they basically highlight that Omicron still continues to use ACE2 because um but I think the way they use that it uses it is slightly different because of the lack of the cleavage
Yeah.
event. And another interesting thing they picked up on was Omicron might have an extended ACE2 host range,
Huh.
which is uh, an interesting thing because it's something that is, I think, still somewhat controversial because there's this one mutation N501Y that we've spotted a lot that is both beneficial in humans and
Hmm.
mice, which means that whenever we see an ACE2 that binds well to mice as well as humans, it could just be because of that mutation. And like, so the assumption is, oh, maybe it's been evolving life in mice, but of course, a mouse scientist could easily come up with the opposite hypothesis, go, oh, it's been evolving in
Uh-huh.
humans. So so it's because it gives benefits to both species. So what they do here is they actually compare this with other spike proteins that have that N501Y
Hmm.
mutation as well, um, and try to compare that, and they find that Omicron is still doing slightly better.
In
And I
binding,
think they do
in binding humanist, no,
in
fold change.
let's see. Yeah, here's like so they tested a bunch of species. So this is the ACE2 data for for the different strains. So first one we've got like this the fruit bat I think
Mhm.
variant. No, not fruit bat, right? Sorry, alpha variant. Uh, so that seems to be that has quite a wide ACE2 range. And
Yeah,
like in I think in that dark gray, it's overlaying the original wild type, the Victoria variant, I believe it was called.
I see.
Um, and so each one that goes down, so beta variant has got a high host range. Gamma variant as well also seems to be doing very well with mouse and rat. And of course, this is just ACE2 binding, so it doesn't necessarily tell us about how well it actually gets into cells and actually infects cells because sometimes it can bind to an ACE2, but it can't make that next step because the cell just isn't very hospitable to them.
Right.
Um, and so you've got like Delta variant, and the Delta variant doesn't seem to be very much more human restricted, much closer to the original. And then Omicron, there's a bit of a boost to mouse binding, so I can sort of see that there is uh, an increased like propensity uh, to bind to other ACE2s, but I can also see why that might not be as much of a consideration as people think it is, because other strains have shown like much
The same.
more
Yeah, yeah,
the
and
same
it
kind of
didn't
variety.
drive any
Yeah,
sort of event, at least that we know of, right? This is in some
yeah,
ways like the hypothesis of like extended host range. Like you really need to see the data in some reservoir or something like that, or some like, I don't know, sentinel animals that you set out in hospitals.
yeah. So they're doing that with white-tailed deer at the moment, as
That's right.
but I don't know about like other strains.
Yeah.
And so it does like also mean that potentially these other variants might have gone through that route. I mean, it might be
Yeah, I
open to
mean,
them,
you know,
but we,
it's kind of
yeah,
a moot point, like, or like in the sense of like knowing what actually happened. Like we only have our hypotheses here, and but yeah, like it definitely having Omicron pop up like this and show the same kind of like extended host range, like it does. I do think that there's definitely uh, one could posit that it would mean that like it had an increased way of getting into reservoirs. Whether or not it actually
Yeah,
has done that, we don't know.
yeah, it's hard to because just because it looks like it's good, there's all these other factors it needs in order to jump into the species,
Hmm.
and
Hmm.
we're not saying that it hasn't done that, but we don't have evidence to
Yeah,
say that. I don't think.
yeah,
Well, at least I haven't seen anything that has really convinced me yet.
yeah. Same.
Anyway, where was I?
Right. This is
Okay,
like in
so
the supplemental section.
yeah, uh, always hate supplementals. I feel like eLife is the one that does it best because supplementals are always
Yeah,
near
you
to the
want to be
data
able to
with it.
flip through the figures and see the supplementals like side by side with them. If you have to scroll all the way down to the bottom of the journal, sometimes it breaks the flow of reading.
Yeah. So yeah, going down. Let's see.
So maybe like figure four.
Let's see. Oh yeah, figure four. That's where we are. So this is
Pick back
again
up
looking
there.
at yeah,
Syncytial
looking at this.
formation.
Yeah, this is actually. I think this is an interesting one because I think they took some of the Omicron like mutations in the furin cleavage site and transferred them over to different strains and to
Hmm.
see. And I think they still found. So what they found was kind of weird, where they if they transferred those mutations to. I'm not actually. I might have got this,
No.
but oh yeah.
Oh yeah.
Uh, so so like things like the P681H that was found in Omicron increases cleavage. I think they had a couple of other ones but then apparently once you have them all together in Omicron, it doesn't do that. So it's very odd.
I mean, it's like cleavage. It does like increase, right? Like this is like I think it's trying to reconcile that idea that it's not. You would think that it would be good at getting in at the surface
Mm.
if it had these mutations, but it's not for some reason. There's something else. The
Yeah.
delta CS is like a larger deletion.
Right. Okay. Maybe that. Yeah.
So there's something else going on in the like you can look at single mutations in Omicron and say that it would be good at getting in on the surface,
Yeah.
or
I
and
mean
forming
that's
syncytia. But that's not what
literally
it
what I
is.
said when I first
Yeah,
saw the sequence.
right.
But yeah, you're right. I mean, this is again circling a hypothesis train. We know that there's something different. We know that these don't work slightly differently, but it's going to take a lot more time for those protein crystallization experiments to happen, for people to solve the structure, and then even more to solve that how it binds to ACE2 or how it how it has how it resists
Yeah, I
furin
don't think we
cleavage.
ever really looked at any, like, I don't remember reviewing data where we saw like the actual interaction between ACE2 and Spike, like dynamically, or I don't know how I don't know how it would be done actually, but like seeing maybe the pre-fusion and the post-fusion, something like that across variants.
Yeah,
That's a difficult piece of data to grab.
yeah. I feel like I might there might be some cryo EM that I might have we might have gone because I feel like
Maybe
that's
there was a
yeah.
paper that we had on the like that I kept in the potentials list that was really old, looking at like some fret studies or something like that. Like I could imagine that would be a methodology that maybe you could use with the variants to be like, oh, look, like in the variants, like two sections of the protein are getting closer than others, and that might
Mm,
be explaining more of the dynamics, right? Because fusion is a dynamic process where
yeah.
the proteins are changing shape in some ways to get it to go. It's not just affinity to binding that is going to affect that.
Yeah. So I think they
So
again
in
they
figure
yeah.
five, yeah, in figure five, they are going across all these different cell types. And again, like the Calu 3 cell type doesn't do well there.
Yeah.
And Keiko 2, that's a gut cell type, right? That's like a intestinal epithelial, I think.
Yeah. Yeah, I mean there's this been because I that is interesting from the perspective of like the idea that SARS can colonize the intestine. Um
Hmm.
yeah,
And then in this paper too, they also use Camostat
they use and
to block.
yeah, Camstat and amphotericin B I think as the endocytosis messer upper of um inhibitor.
Yeah.
That's what I wanted to say.
And this is in primary cells, so yeah, those human nasal epithelial cultures. And yeah, Camostat has a really big effect on Delta, but not, it does have some effect on Omicron, just like not as much.
Yeah, so that fusion is still a thing for Omicron. It's just not as
Yep.
much.
Yep.
So yeah, this kind of like paints a picture that Omicron is entering cells in a different way, and that it is infecting the upper respiratory tract in
Yeah.
the nasal tract much more.
In
Which
Figure 7, they throw a nice model for like a visual thing. If people are like visual, I think that's like one
yeah,
of my favorite things about the way journal publishing has gone. Like those graphical abstracts, I first saw them in like Cell or whatever, but they're really useful. Just like as like a visual summary of sort of some of the data that's being discussed inside the paper.
yeah, and one of the things that they point out in this paper, I think this is a paper we covered like a while ago called Moose at all, because I remember I covering because it was a single cell transcriptomics paper, which is like my jam. And they
Sorry, what
and
did you call it? What did you think it was
Moose
called,
et
that paper?
al.
Musetto?
MUUS. So where they
Okay, sure.
cataloged all the like cells with ACE2 and T imperioris 2 to
Ah,
try and
yes,
figure out which one
I
SARS can
remember
go
this.
into. And
Yeah.
like, I think the point that this paper points out is that lots of cells have ACE2 but don't have TMPRSS2, and a lot of them are in the respiratory tract. So so that means that Omicron has a much broader like amount of cells that it can infect. So even if it
Hmm.
is slightly less good at infecting those cells, it can still start off an infection faster.
Yep. Yep. Yeah, so that would be the way that they're tying this observation of the biochemical to the outcome of the infection that we're seeing.
Yeah. So I think that this is a really good start on understanding how Omicron works and how it's quite different from the other variants. Um,
Totally.
but yeah, still there's a lot more work that needs to be done because at the moment we've drawn like a circle. We haven't like hit the nail on the head yet. There's still quite a big circle around, and there's more questions that we need to have answered. But I think this week this has been really interesting news. I think,
Yeah,
and yeah,
absolutely. I think it's tied together some of the observations that we saw last week into a real story.
yeah, and that's like both great, but also kind of perilous because science and stories don't necessarily mix together. Sometimes it could be an alternative, much more complex and weird explanation for things. So
Yeah.
that's me hedging, by the way. I'm hedging to so that just in case.
Okay, but like shifting gears a little bit, still Omicron, but now asking the question, there's also Delta out there, right? And we saw that Delta has like a more fusogenic, right? Like that's maybe part of its success has been just like how great it fuses. So like you get a little bit and it can spread quite easily. So the question that the authors are asking in our next paper called Omicron infection enhances neutralizing immunity against the Delta variant is, yeah, let's look at Omicron cases. Let's look at serum from Omicron infected patients and what's its effect on Delta. And they do that by enrolling a couple of people, taking their serum, and then testing that in a cell culture model.
Yeah, so again, an antibody-focused study, but it could still yield some important information. Yeah, this was interesting to me because because there's this. I mean, I was this is kind of interesting because the Omicron is so different from the Delta variant. So trying to see whether there's like that kind of crossover there and also whether it goes both ways.
Yes, yeah, yeah. I mean, in some ways, I think the idea is that it probably doesn't go both ways, right? Because we've had this surge in Omicron, but we don't know until we test.
Yeah, because you think they wouldn't go both ways because of the fact that Omicron is much better at infecting people who've been vaccinated and or
Hmm.
people who've already had the disease.
Yeah.
So what seems to be fine is this is a weird like kind of result that I'm not sure I can really logically explain.
Okay, so like what's happening here? They have I can't even see what we're showing people on the screen.
Oh, sorry.
Actually, I have my
We're
Zotero.
showing
No, I have my Zotero open looking
figure
at
one. Yeah.
some other papers. Yeah, so here we have like they enrolled people, they took their serum, and then they looked at this neutralizing how much serum it took dilution wise to 50%, right, reduction of infectious foci is that's
Yeah.
FLNT. And so yeah, Omicron Delta. in both cases they see in follow-up you get more. So like there's that idea that like after the event, all these people were enrolled like 4 days approximately from I think a median of 4 days from their infectious event where they were submitted to the hospital PCR
Oh,
positive and all
14
that.
days, I think. Oh, wait, no, wait, hold on, wait. Measured
No,
from.
and then follow-up happened afterwards. That could be 14. I think the median's 14 follow-up.
Okay, yeah, that makes.
Yeah.
No, you're right.
So
Right.
then, so that's like that, and this is something that we know from before as well, and that we know from vaccination studies, right? Over time, like you do, after that event of being exposed to the antigen, like later on, you have more antibodies to stuff. But then what's interesting is it's this 50 it's the C, right? Figure C is interesting because here they're looking at like with Omicron serum, right? I think at the at the follow-up time point. Yeah, this is all the
Yes.
follow-up time point now. So at the point where you have the highest antibody titers, right, to do something with, right? Like what's that? How well does it neutralize Omicron versus Delta? And you can see that it does have some effect on Delta. In some cases, right, it has effect on Delta.
Yeah, so the orange line's
Yeah.
unvaccinated. So so some of the unvaccinated are doing worse, but most of the people who are vaccinated tend to be doing better against Delta.
But again, I mean, that's kind of interesting too, because, right, we've said before that the vaccine gives you like a slightly different type of immunity, right? It doesn't, it's not a natural infection. So like it tickles our immune system in a different way. And that's helpful in this case, it seems like. Again, it's a kind of a small sample size to draw those types of conclusions, but it is helpful in this case because, yeah, it's sort of working together with your Omicron infection. My goodness. Unfortunately, still you don't want to be infected, right?
Yeah.
And
See what other because
yeah, and that's
yeah.
sort of what it's telling us here. That's the big figure. Then there's a supplemental where they're just saying like, yeah, Omicron is still evading vaccines, or yes, Omicron evades immunity better than from OG, better than OG.
Yeah, from what we're going to refer to as the original genotype, the OG. Yeah. Um, and
Yeah,
so
so it's kind of,
next,
I guess it's supposed to be. I feel like the thrust of this paper, reading it, yeah, it's quite short, so it's sort of trying to say like, what's the future, right? Given that Omicron, like I heard in the news that, like South Africa, like says that the peak of Omicron is like over now, or like they're in the peak of it, and like they expect cases to go down. So like, what does that mean for, right, Delta, which is still out there, right, and other infections, or maybe another variant, right? It's trying to I guess it kind of weakly says that like we are getting some immunity from being infected like this.
yeah, I think the worry with Omicron is because it was so different from the other variants that perhaps it wouldn't confer the same level of protection because it seemed like it was overriding the protection conferred by the other
Right.
variants. So this but I think this study there is that confounded that you are catching things at the peak of antibodies like production, and so it remains to be seen whether there's like a waning effect
Yeah.
that we've heard of with vaccines and other infections. Uh, so yeah, this is. I mean, I'm not going to discount good news, but it's still like I wouldn't like bet the farm on it.
Right. Yeah, I think actually what's emerged, and I think this is my segue into the next paper, is that like this idea of antibodies, it is like just for that initial infection and transmissibility, right? Omicron's like super transmissible in some ways because it like overcomes some base level of antibodies we've had, but there's still the long term, like once you get infected, like your body goes into production mode, right, and starts making more antibodies. And so like you might become infectious and have some symptoms, but then you control it afterwards through a combination of, and that's going to be reliant on T cells and memory responses that happen during infection.
Oh, funny you should be talking about that because our next paper is SARS-CoV-2 spike T cell responses induced upon vaccination or infection remain robust against Omicron. So yeah, this is I think I've been harping on a lot about how important T cells are. And this paper actually, so last time we had a T cell paper, it was focused on multiple sclerosis patients,
Yep.
and this one it takes a much more like larger look at different people. So let's see about this. So yeah, they looked at the ability of T cells to react to spike, and so I think it's they so what they so essentially this is another paper where they show slices of spike protein peptides to T cells to see whether it makes the T cells angry or not.
Yes. Yes.
Um,
Yeah, and they can show either the pieces that are more similar to they're calling it ancestral strain here instead of OG. I guess like the more sophisticated terminology versus or Omicron, and see if it still makes the T cells angry, and it does.
yep, it does. So that's good news because and what they said is like there's like only like a 70 like there is a slight decline that they see because you're going to see that with any kind of strain, but it's only about between 70 to 80%
Hmm.
of the CD8. So CD4 the T helper cells and CD8 the cytotoxic T cells.
Hmm.
So CD4 are the brains, CD8s are the fists. Um,
Yeah.
yeah. And I think that they found like in terms of like cross reactivity as well that there are still like T cells that are cross reactive to most of the
Hmm.
variants. Um,
And so this
I
this
think
actually
like
this is to me this data is like because they're looking at this in human samples, and like we're asking how does this relate to like the overall picture of what we know about SARS-CoV-2 and how it works with vaccines is that we still see a significant reduction in serious hospitalizations, right, from vaccinated
mm,
individuals and SARS-CoV-2. And like here could be that reason, right? Even though it's breaking through your antibodies, you still have other cellular mechanisms that confer protection from the worst outcome of infection.
yeah, I think that that's an important point. Like so antibody responses might go down, but that doesn't necessarily mean that your immune system is dispit. So because we've seen like lots of studies where Omicron has like performed where antibodies are really really bad at neutralizing Omicron,
Yeah, yeah.
but T cells seem to be there as a backup.
Hmm.
Um, so I think that that's an important point. So that when we see kind of studies where they say that all the immunity has disappeared based off of antibody data, we need to be kind of aware that there's other parts of the immune system that will still
Yeah.
react, and
Ju-
and so
it just
it's
tempers. It's like it's a very like immune system such a complicated picture, right? And so it just
mm,
tempers that maybe that initial fear of like, oh my God, it's getting it's getting through one layer of defenses. But like, okay, there's some other layers of defenses. So like, really, the question becomes like, what does it mean to overcome antibody-based immunity, right? Oh, it means like spreading means breakthrough infections, right? But then hopefully people aren't dying.
mm,
But with more breakthrough infections, it does mean higher case numbers. And if everything's just a percentage of stuff, right? A percentage of a lot of case numbers could end up resulting in a percentage of lots of people going to the hospital. So, yeah, definitely not over, right? Like we're still so much in the
yeah,
pandemic. Yeah.
right. And I think that brings us to the end of the Omicron part of our coverage.
Our
So we're
final,
just going to be
no,
doing a
our
look.
final Omicron paper. Oh, I go right. This is just SARS-CoV-2.
Yeah, so the next one is just
Yeah.
SARS-CoV-2. But it's an important one. I think you'd want to. So if you're just here for Omicron, you might want to stay for the next one because this could be quite an important one because this
Yeah.
is SARS-CoV-2 infection and persistence throughout the human body and brain.
Hmm.
So
So
this was like sent,
autopsies.
yeah, via autopsies.
Hmm.
So
Human
uh,
tissue. Very exciting.
yeah, human tissue, human autopsy. So we've gotten quite a lot of important data through human autopsies. Um, And so this is based on a population of 44 individuals who died from COVID-19. And the interesting thing about this paper is actually the length of the disease, because some of these people had like COVID-19 for 230 days. So these are people who are so. And another interesting thing is that there's one of these people. I think like they had. So they mentioned they had like two cases where they'd actually resolved the COVID-19 infection and then were later died of other causes. And upon the autopsy, they found that COVID-19 was still in their tissues somewhere. And I think that's
Yes.
the
Well, this is that long COVID question, right? Like for some people do experience symptoms, right, that go a bit further than an initial infection, and this could be a part of that trail to go down.
yeah. So they found it ended up so I think I should emphasize that quite a lot of these patients were had very severe COVID-19. So let me so I need to be careful here with what I show because I'm not entirely sure that about their but I want to like so I'm blanking out some of the patient data because some of these
Mm,
patients had rare diseases that could make them very identifiable. So I want to
gotcha.
be, and I'm not sure whether the authors had that level. Well, they what you usually look for papers like this is like patients that provide consent to publish.
Right.
They provide consent to autopsy, which isn't necessarily the same thing,
Ah,
and
yeah,
I'm very wary because there have been like some court cases where patients have read about the deaths of their loved ones through an academic paper, and
yeah.
they didn't know that, they didn't expect that to turn up. So
Right.
I want to be kind of sensitive to that.
That's
Um,
that's that's probably for the best. I also I do want to talk about
so,
Minref's comment. Like some other scientist says, like T cells mediating severity is not doesn't have any data behind it. Maybe I guess that's can't find a single bit of meat.
oh wait,
But I thought we read a paper that said that
we
T cell epitope was linked to severity.
yeah, there was like so there's that paper that looked at the T cell epitopes for the polymerase gene that was linked to asymptomatic transmission. They also had
Right.
some other T cell paper because I know
Yeah.
we've had a
So it
couple of
might
T
not
cell
be a
papers
well
where they
developed. It might not be a well developed sort of trope or like hypothesis in this sense. But I think we have seen some inklings that there could be a signal for severe disease in that. And again, that's just like a marker in some
but
ways.
yeah,
Like so yeah, I'm not sure of it. Yeah, but
I
it also
mean
makes sense from my theory of or like my understanding of the immune system that like there should be something we have at its most basic level, there still has to be an immune response to go down the severity path to differentiate
yeah.
between the two severities.
Sorry, I so they've got some like actually what quite good like pictures on this. So I'll unblank the pictures because they show like the SARS-CoV-2 in different like organisms and
Yep.
like it's not
So they're
organs
using
organs.
in situ hybridization for these types of pictures. So what that means is that they have the tissues from the autopsies, and then
Mm,
they have probes that recognize some piece of RNA in SARS-CoV-2. So then you wash the slices of tissue with your probes, and then the probes will stick if there's any residual SARS-CoV-2 RNA in those particular cells, and then you can develop the image and visualize what you're seeing.
yeah, so I think the brown bits are the SARS-CoV-2 and the
Yeah.
other bits are the counter stain to make sure that you can see the other cells and put them in
Hmm.
context. So you got
Hmm.
uh, unfortunately my histopathology is not very good, so I can't identify all these cell types
Yeah, I
immediately.
don't
Just
see the I don't know what the structures are. I just see
I
blue and brown.
yeah so yeah
But yeah, you just
uh,
passed the
but yeah
lung, and then
to
the next one
yeah
is the
but to
cerebellum.
yeah so sorry to add to your point about the T cells, I feel like that there are some papers that are saying that T cells have been linked to better outcomes, but then I do also understand that there might be like other papers linking certain antibodies or B cell types to certain outcomes as well. So
Hmm.
I can understand why someone might go to the perspective of saying, okay, well that isn't conclusive evidence because you could say that about
Right.
any cell type.
Yeah. Totally. Yeah.
So yeah,
We don't
uh,
have, I guess maybe what's important to say is we don't have a single understanding of what mediates severe disease versus non-severe disease.
yeah.
But there's a good there it is. I think we can say that like immune response, right, triggered by uncontrolled viral replication is what gives us that severe case. So
Mm.
anything that diminishes that could be antibodies, but it could be like CD8 T cells doing their thing. Maybe that's the thing. We've
Yeah,
never seen CD8 T cells directly relinked to like some outcome of disease, right? And like that was
yeah.
like, those are the effector T cells.
Oh yeah, and there's another cool thing they found about like genetic diversity across the compartments. So the idea that when a SARS-CoV-2 goes into a body compartment and stays there for a long time, it evolves. And they actually did some analysis on on SARS-CoV-2 in different. So I thought that was quite an
Oh, in this
interesting part of
paper.
the
Yeah,
paper as
yeah, yeah.
well.
No, I really
Yeah.
like this
So
data
the
set that they brought together
yeah.
for tissue types.
But yeah, it's really interesting because we have had this story about about SARS-CoV-2 evolving during convalescent
Mhm.
infection. But of course, that like when a virus or anything goes like septic and it enters your bloodstream and it can go anywhere. And I think the one limiting factor for this kind of research is the fact that researchers we can't sample everywhere at the same time.
Yeah,
So you have to look at the most likely places, which means that there are blind spots all over the place because simply we can't like sample every single cell
exactly,
in like
exactly. But in this instance where they I guess they have a lot of time from the autopsy, right? They
yeah.
they can really dive into that. Can
Well,
we show this figure extended data figure 2?
let's see, and yeah, I think we can share that.
Yeah,
Let me
there's no
see.
I don't see any
Yeah, I
patient
think
sort of info there. It's like a
yeah.
big table.
So if
Yeah,
there's
I don't you're gonna have to turn it
yeah,
around or make it smaller or something. But the idea here is that
but
every row, in this view, every row, right, that you're looking at, it goes from actually probably from the bottom to the top is like a different patient. And then in every column, they have all the different tissue types that they looked at. And
yeah,
the colors that they're giving is like how much they saw RNA, how much they saw staining for the RNA, I think. Yeah, average copies.
yeah,
And so like you really get this
yeah.
sense
This
of
is
so that first column chunk is respiratory. So you can see like going up that
mm,
first column chunk, there's like lots of red, right? Like the respiratory tissues, yeah, it's definitely there. SARS-CoV-2, respiratory virus,
mm,
we see tons of that. But like let's go down. Next is cardiovascular, right? And we're still seeing quite a bit in the
mm,
cardiovascular column. Lymphoid, in that's in immune cells. Gastrointestinal, right? We're seeing a whole bunch of different cases
yeah,
of gastrointestinal. Renal, I've actually I saw some papers go by me too that like we're talking about involvement in the kidneys. I
yeah,
can't read the next one upside down. Then
yeah.
is skin, muscle, skin, and adipose. Then is ocular, and then that final one is
Hmm.
central nervous system. And so what I wanted to
Yeah.
point out to people here in the central nervous system one, it's not like scattered over all these different patients. It's select patients have like a lot of cerebral CNS staining. Yeah,
Yeah.
right. Like it's very
Now
clear there
that
that it's like it's not
yeah.
like oh in some regions it's like once it gets in someone's brain then like that person like gets it sort of spread across. And so I don't really know how this plays out. Like this is just very observational. It's not driven by statistics here, but yeah, like this is something to consider, right? When people are talking about long COVID and we don't know who's susceptible to this particular invasion, right? Like we don't know the clinical histories of these. This might be something that someone could discuss in a case study related paper that gets published.
Yeah, I mean, it isn't because I think like sepsis is quite interesting, like in terms of like things getting into the blood system and entering different cellular compartment, different like organ compartments,
Yeah.
and the nervous system is like a compartment of its own slightly because of the blood brain barrier.
Mm.
So I feel like the so once it enters like the nervous system circulation, I feel like there's like it overcomes that step and can go into other places.
Hmm.
But that's just me speculating.
Yeah. And so yeah, that's this paper. I think we're good. I'm good to move on.
Yeah. Yeah, I'm good to move on as well. So yeah, there's I mean, I should highlight that a lot of these patients did have comorbidities, but I think that there is like a point here that what's it? Uh, oh no, I lost my point. So let's move on to the next paper.
Well, I'll springboard off of it. I mean, one point about having comorbidities in this group is that like, that's an indicator of severe disease, right? So maybe in like perfectly healthy individuals, right? Like SARS-CoV-2 is just going to be like in your lungs, but that's not many people around, right? Many people around the world have comorbidities of one sort or another. And like if we are thinking from the public
Yeah,
health perspective of keeping folks safe, then like controlling spread is important, right? Even if it's not for you. And like that's something that, it's even important with the flu. I feel like this whole pandemic has just like tuning the ways we talk about infectious disease and like what our role
mm,
is as sort of like interacting human beings in controlling spread.
yeah. I remember what my point was. So even in the patients who are asymptomatic and didn't die of SARS-CoV-2 and like had the disease resolve where they found it in their tissues, the disease they had was quite severe. So some of them were on like intubated and had like so I think the idea is like that there is a point in infection, especially in severe infection, where SARS-CoV-2 can go septic, and it can and so septic is based.
Yeah,
Well, my
in
interpretation of
circulation.
sepsis is not necessarily you get into circulation, and then once you resolve the disease, like in your lungs or wherever, that SARS-CoV-2 might not go away.
Right.
So that's one of the thoughts that this paper like instilled in me. But again, like they saw it in I think 2 patients, and it'd be interesting to see like whether we could replicate this or whether what can be expanded upon in this research.
Yeah. Yeah. Like in 2 patients, but then across all of these, we don't know which ones they match up to. Yeah, the researchers have to go in, right? Because these are like they could potentially have access to data that could tell a story that would be helpful in directing sort of future hypotheses about what is SARS-CoV-2 doing in these tissues, and how
Yeah,
did it
like
get there?
yeah, I mean we've still got like a lot of questions to ask about like what SARS-CoV-2 does in the brain,
Yeah.
which brings us to our next paper, which is Microglia do not restrict SARS-CoV-2 replication following infection of the central nervous system of K18 ACE2 transgenic mice.
Yeah, so here it's a mouse study, but they've been humanized appropriately so that they can be used as a SARS-CoV-2 model, and they infect them, and they see that they infect these mice in their infectious model. They see that what we've always seen goes into the lungs. I'm trying to scroll down to some of these images here for myself.
Yeah,
It's one of
I'm
these like
trying.
preprint style things. I think you'll have to go to the PDF. Yeah.
Oh yeah, because essentially what happens is like when a journal receives something, it takes a while for the production department to make it okay for the web page. So we're at
Yep.
that point where there's someone who's trying to retype this all to add onto the web page and format it. So
It's page 31 is where the figures start.
okay,
And
excellent.
of course they're without legend because those are typeset separate. But anyway,
Yeah.
so they can take brain slices from these mice, and they can stain for neurons, and then you can stain for SARS-CoV-2, and you can see that yes, they're in the neurons of these mice 48 hours post-infection. Um, you can talk about like some of the genes that are turned on in this
Yeah.
case, immune genes that are getting turned on because the brain does have its own sort of separate immune system, mostly instigated through these microglia cells,
Hmm.
and so they do some staining to see like in the next figure they do some staining to see like I guess sections of the brain that are getting infected and what that looks like. um, what that looks like in terms of immune markers that are being expressed in those areas. They can see, yeah, CD8 cells are getting into those zones. Yeah, so there are microglia get activated in this sense
Yeah.
to suppress SARS-CoV-2. Um, okay, yes, then they have some bigger brain sections, and then
Yeah.
where did they?
Very. Yeah. Very like expressive brain sections where they look at and see. I think I I don't have the. But yeah, it looks.
Yeah, I know it's hard to get to the figures like or the legends right. They're seeing SARS-CoV-2 in the brain. Like
Yeah.
this is the most of the points that we're getting through. Let's keep
Yeah.
scrolling.
I think the
Scroll
pink
down
stuff
to
is the SARS-CoV-2, but I don't know.
yeah, it is. It is. I mean, certainly in this figure, it is.
Yeah. So yeah, we're looking at it in various like aspects of the mouse brain.
Yep.
Uh, again,
And
it's very hard.
in
Yeah.
the next figure, they stain for what we would call the microglia cells,
Right.
MAC2, I think. So like they see that like so there's infected cells, and then there's also the immune cells that are going to those locations.
Mm.
Okay, and then the final figure, which is the interesting finding here, is that there's like some sort of mouse chow that you can give to mice that suppresses their ability to activate microglial cells.
Oh
And so
wow.
they feed the mice this chow, and they actually don't see any difference in what's going on in the brain, or in like the initial weight outcomes, or in the amount of spike expression that they're seeing in brain slices. So that's
So
A and B.
right, I see. So like, uh, so the SARS-CoV-2 is there, but it's not necessarily like causing what you'd think of as a traditional disease.
I'm
I
not sure.
guess.
SARS-CoV-2 is there.
Oh
Yeah,
oh
it's replicating at some amount. It recruits microglial cells to that surface, but the microglial cells are not important in controlling anything.
Okay,
Yeah.
so that's an interesting data point.
Yeah, that's the data point being expressed here.
Yeah. So I think
So
that
basically,
brings
it's
us.
in some
Yeah.
ways it's a very complicated negative result, but it says like maybe we could think about other things in terms of like what would govern the outcome, or maybe it doesn't really get well controlled. I mean, in the autopsy samples we saw, it wasn't like single tissue areas
Mm.
that they saw; it was across the whole brain. Maybe that's just what it is. Like if you are somebody who has the unfortunate right fate that SARS-CoV-2 is able to cross the blood-brain barrier, or I don't know, I don't know what the way it gets into the brain. Does it go like in the neurons, olfactory neurons, or something like that? Then there is no good way to control it there, but it also doesn't cause that severe of a illness. I'm not sure.
Well,
There's nothing really to say illness here. It didn't look.
yeah, I think the thing about microglia is that they're in a very sensitive area, and if we, and the thing is like reacting to things can cause more damage.
Yeah.
So it's almost like it's like jazz, where the notes you don't play can be as important as the
Yeah.
notes you do play. Whereas so they just because they aren't necessarily causing SARS-CoV-2 infection doesn't mean they're not doing other things to limit damage to the brain.
Right, right. That's a good point. Yeah, they could be talking to cells saying like, "Don't freak out. Like, there's something
Yeah,
strange inside of you, but it's not time to do apoptosis yet."
yeah. I mean, if the SARS-CoV-2 isn't killing off the brain cells by itself, then it isn't actually causing harm in that, causing like a specific like kind of like direct harm in
Yeah,
the brain, then the immune system might not do anything. It might be doing other things that are more subtle and might affect the mouse's behavior, but that's very complex research to do.
right,
So it's
right.
so I don't think we'd get solid answers on that for a while.
But that's the takeaway here. A small glimpse into what might be happening. Again, this is a mouse model, but like what might be happening in human SARS-CoV-2 infection, particular as it relates to immune responses in the brain.
Right. And I think that brings us to the end of our SARS-CoV-2 coverage.
Yes.
And now we've got like some interesting papers from the other world, from the world of microbiology.
Yeah, just two papers. And the first one we have is virulence perforate the pathogen containing vacuole to signal efferocytosis. So actually, I didn't even know what efferocytosis was. That's one of the reasons I'm like,
Yep.
I guess we should pick this because I just want to read the definition of it. And the graphical abstract, I think, does a good job in sort of pointing to people what this is. Like phagocytosis is when like the cells they want to eat these extracellular bacteria, put them in a very specific type of vacuole where they're going to get killed. Um, but efferocytosis is just when neutrophils are cleaning up, I guess, broken cells.
Yeah,
um,
because cells are filled with all sorts of like crap that you don't want to get out there. So they've got like all sorts of enzymes that break down proteins and stuff. And with apoptosis, you don't want the cell to burst and release a bunch of like bad like all the enzymes it uses to break down its food. You need to have like a controlled demolition. As part of that controlled demolition, a phagocyte comes along and eats the dying cell.
mhm.
So
Oh yes,
that's
I was just looking at Minref's comment. Yeah, it's something about like how the news being disseminated around T cells. Yeah, so and I think what to think about in Salmonella's case is Salmonella grows in one type of immune cell, macrophages. They have like a
mm,
secretion system that allows them to survive in a macrophage um, vesicle, right? Like from being phagocytosed by a macrophage, but they don't really have ways to survive inside of, I guess, being phagocytosed by a neutrophil, or maybe not as many ways. And so the idea behind this lifestyle for Salmonella is that once like they're in a cell that dies, like they want to have, I guess, time to figure out what to do when they get ingested by a neutrophil, and maybe maybe specific cell factors, virulence factors that they have help them get taken up through that pathway rather than some other pathway where they would find themselves digested.
yeah. So
So
they
yeah, scrolling through the figure. Oh, sorry, you wanna?
no. I was gonna like say this is interesting because like they looking at a type 3 secretion system to look at
Hmm.
like because I think there's some instances where like a certain like bacterial like bacterial protein, I think flagellin, gets translocated out accidentally during its infection cycle, and that causes the cell to undergo like apoptosis or pyroptosis, I think they call it, which is
Yep.
like that, but in reaction to like a disease. And
Hmm.
that overreaction is not good for the salmonella. So this kind of studies like looks into kind of, I think there's like a paradox they found in there, but I couldn't quite. This study was, I was still like half of COVID research, so I couldn't quite
Yeah,
get
yeah.
the.
This is also a very long. This is a very long study. Their story exactly, just that. Yeah, type 3 secretion is used to send virulence factors into things. So salmonella, and I think what
Mm,
they do is they're able to remove when they remove the ability to secrete something, they lose this ability to get efferocytosed as opposed to being phagocytosed by neutrophils. So like figure
right, right.
4, I think in figure 4 they have um, 4I, they have like two graphs where they looked at yeah, phagocytosis versus efferocytosis. They have some expression. I see they're looking at expression. Do like no, I can't see. Anyways, something would be interesting. I mean, like I love these. I didn't get a good chance to dig into this as much as I normally. But yeah, like this is just like one of those many stories. Like when you really dive into the life cycle of a bacteria, and you think like all the different environments that it has to encounter, especially when it's inside of our bodies, right? It's encountering like all these different cells that are trying to kill it, or things that don't want it around, but it has to grow. It has to find that place to grow. And virulence factors, and specifically ones that are secreted, are like really important factors in changing changing the types of environments it ends up encountering.
Yeah. I don't really have much more to say about
Okay,
this.
so let's pop over to the last one, which is kind of a big data analysis of like publicly available data, systematic evaluation of horizontal gene transfer between eukaryotes and viruses.
Yeah, this is interesting because I think we've like seen a lot of like gene state change between bacteria and viruses a lot, but I don't really like see that often with
Yeah, and
eukaryotes.
they pointed out in their description of this paper that there are there are actually a bunch of examples where we know that viruses did something to eukaryotes, like some placenta protein, right? Placental proteins, salivary amylase. There's all these
Mm.
examples, but no one's ever gone through and just like, right, we're in the age of big data now, right? Like we can just take all the data that we have and try to look for those events and then try to find patterns in them, and that's what they end up doing here. I think also I think there's also sorry like now I'm all these examples are flowing in my head. There are also examples of eukaryotic cells taking viral genes and then using them as like immunity against other viruses or something like that. Um,
Yes, I do know examples of that.
Yeah, I think that's like Tetherin might be that one for HIV control. Anyways, there's lots of different ways, and then on the virus side, it is known that like viruses have picked up like transcription factors or like things to control cell cycle, because I mean they need to control the cell cycle
Yeah,
because they're replicating in cells. What better tools to do that than like a cell control gene from the host that they have? And so the way that the way that we can find horizontal gene transfer is you make an evolutionary tree of a specific protein, and then if you know then you look at like the ends of those trees, and if you find like one like if you find a gene that is a sort like all the things around it are closely related. Like they're all from, in this case, they're all from eukaryote or they're all from viruses. But then you find one in that mix that is assigned to the other group. Then you'd be like, oh, this seems like it's something that might have hopped, right? Because its its evolutionary ancestors are not from other viruses, but rather from the other kingdom, and vice versa. So I think figure 1A, that's the sort of the the schematic of what people are
Right.
seeing.
Yeah.
So this is very big data, right? Like they create trees for every protein family. Um,
Uh, yeah.
they
Uh,
try to
and
assign
we're
these
definitely
effects.
seeing like yeah.
Yeah, and then so
And yeah,
A2 they
it's
give you the
interesting.
numbers,
Yeah.
and they know that there are types of artifacts that occur when you create trees that could make something look like it's horizontal gene transfer, but it isn't quite. So they do have like well supported versus weakly supported based on what the researchers know as potential artifacts from tree making, or artifact itself. And so but I because I trust these researchers, right, I haven't dug into this paper as much as I could have, but like if we trust these researchers and we say let's just like they did that work and like let's trust the well supported, there's still like there's still a bunch of examples that they're able to pull out in this data.
Yeah, I mean, just looking at this tree, I'm seeing like a lot of like for single cell or like eukaryotes like yeasts, alga, and all that. It's
Mm-hmm.
all the like integrations seem to be much more recent,
Mm-hmm.
whereas like with the multicellular eukaryotes, you see like much more ancient, like as if we've seen these trends. So very few are like very recent
Yeah.
from recent common ancestors.
Mm-hmm, mm-hmm. Yeah, and that was one of the things
But
that they
that's like
mentioned in their results, like that idea that like it happens like a long time ago in multicellular. They first of all, there's more representation from unicellular than multicellular in the transmission events, and yeah, that's probably because like unicellular things they don't have to worry about the Weissman barrier that's mentioned in this paper, right? Like
yeah,
they're just the same cell that becomes they don't have a separate germ line that sort of like protects their genome from changes like this. They do mention
yeah,
that there are not that many retroviral assemblies that are separate from the host. Um,
mm,
in their data set. So there might be more instances, right, of multicellular gene transfers, but like there's no way to annotate what the viral gene is in that case because like there's no separate assemblies of retroviruses. Um, so that may
right.
be something that drives the pattern that we're seeing there. Yeah, and then they try to just find the function of some of these things, right? Like they want to see like the function of genes that go from viruses to eukaryotes or to eukaryotes from viruses. And I got kind of lost in this. I mean, like they obviously have them in graph form, but yeah, there are many different functions they
Yeah,
can have. I
it's
think I picked up on like glucotransferases or something. Oh yeah, it's like a subtitle. Viral derived
yeah,
glycosyl transferases. So viruses shuffle around some of these glycosyl transferases, I guess, and that it's a place that you can evolve new things from. And that might be an interesting function that viruses play in overall evolution.
yeah. I mean, it is interesting to see like the role of viruses across like the lives of other organisms
Mm-hmm.
and how like how a lot of like the genetic diversity because I think like what they said one thing they said was like often like an integration of a virus gene like is linked to like a increase in diversity and a new function for like that organism.
Yeah,
So there are opportunities that organisms are given because of a viral infection, and
absolutely.
you never really think of it like that.
Yeah, I mean it's great. Like I'm all for this like Gaia hypothesis thing, and like we're all one big organism, and viruses I think really have like show us some real stories, right, that have occurred in evolutionary history where that's the case.
Yeah. So I think that brings us to our show's
Yeah,
end.
absolutely.
It's been
Yeah, join us
yeah,
next
this
week.
got some really good
Yeah, for
stories. Yeah.
more cool
Sorry.
microbiology news.
Yeah, and we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get everything right. Science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the
Yep, I
comments.
totally agree. You can reach out to us over Twitter at MicroTWJC. We both believe that peer review is a process that we can all participate in, and so we've hoped that you've had a good time listening to us ramble about microbiology today. And thank you to everyone who's been commenting in the comments.
Yeah, again, it's been a pleasure chatting with you, Danny, and so thank you again to all the
Yes.
commenters. It's been great.
Same here, Fuzz. Bye.
Tune in next week for more microbiology content. Until then.